All right, welcome to our next microbiology lecture. Our final lecture looking at prokaryotic cells and the details that we want to get into in this class. Expanding on our basic idea of the prokaryotic cell and pointing out the differences in many cases between gram-negative, gram-positive bacteria as well as the archaea. So we've already talked about plasma membrane differences, we talked about details in the cell wall. In this final lecture, I want to address external structures and then some internal structures. This will be more of a list-like address of these structures because a lot of them we're already familiar with, especially when we get to internal structures. So let's get started with our external structures. We're going to look at the capsules, the slime molds, the slime layers, and then flagella and pili. So glycocalyces, or singular glycocalyx, is basically the capsule. Okay, so you have this glyco, sugar, calyx means kind of shell, or I think of it from a botany point of view, kind of a whorl or a circle of things that encloses the emerging flower. But like that's where the word comes from. So we're going to be surrounding the cell with this gel. So a gel-like layer outside the cell, we think about it as protection, also as kind of gluey, attaching to surfaces. The capsules are going to be distinct and gelatinous, whereas slime layers are diffuse and irregular. And that's as good as it gets in distinguishing between the two. Now, when you have a species that has a significant capsule or slime layer, those colonies are going to appear moist and glistening, right? So again, we've learned that as we isolate bacteria and grow them in the lab for those that we can, the colony morphology can be diagnostic, and we can learn more about that as we get exposure to them and differences. We're like, oh look, this one's got some, it's shiny. It probably has a significant slime layer or capsule, right? Now the chemical composition depends on the species, so that's going to vary significantly. Great review that I found, bacterial capsules, occurrence, mechanisms, and functions this year, 2024. There's the link for that if you want to check that out. But this is one of the figures from it, and I've got a chart from it in the next slide. But it kind of just shows you the importance of how, again, this most external component that the cell is secreting is going to impact how it interacts with other organisms and its environment. So talk about adherence, immune evasion, penetrating the blood-brain barrier, and antibiotic resistance were four that this particular slide or figure in the paper looked at. And then here's a chart that shows you capsular-related bacterial diseases, right? So looking at E. coli, Pseudomonas aeruginosa, Klebsiella pneumoniae, like most of these we'll have exposure to in the lab. So not all of these are going to make you sick, but the strains that have these particular capsule types are going to contribute to the diseases that are associated with them. So we want to understand that all of the components of this cell are going to have an influence on how they interact and potentially how they can cause disease. So what could be a whole nother lecture in and of itself, and something that is of personal interest to me, which I try to read more about, are biofilms. So when you think biofilms, the most common example is think plaque on your teeth, right? And so I think that's a really powerful example because it is when glycocalyces come together. So they can be from a single species as bacteria kind of get together and this gooey layer outside them kind of all comes together. But what's more important and cool about biofilms is that it's oftentimes multiple species of bacteria that are kind of cooperating. And it's almost in some cases, like little cities. There's organization. So it's really interesting stuff. But we want to talk about this EPS, this extracellular polymeric substances, as kind of the general term for this biofilm. Now, if you have ever been to the dentist to get your teeth cleaned, and I know people in this class are sometimes interested in dental hygiene, that's what you're going to be dealing with every day. And what's the instrument they pick up when they want to clean your teeth? from plaque. It's like the metal hook, right? Or they have that like really powerful vibrating jet of water, right? The point here is that biofilms are very effective at protecting those bacteria. And the damage they can cause, but also then the types of chemistry that are associated with biofilms is really, really interesting. So just again, be aware of them and their ability to protect colonies of bacteria, but also then populations, almost like little ecosystems of bacteria as well. The next component that is external on prokaryotes that I want to talk about are the flagella. So again, not every bacterial cell will have a flagellum or plural flagella. And we want to really emphasize the idea that eukaryotes can have flagella as well, but the structure is fundamentally different. It's engineered differently. It's made of tubulin. Um, our microtubules are a component, and like we just don't see that structure at all built the same in prokaryotes. And I'll show you the next slide that'll kind of emphasize that. And how it generates power for motion is different. So whereas bacterial flagella is going to be powered by the proton motive force, again, just like we power ATP synthase with protons moving across it to make ATP kind of spins, we're going to see the same thing in the bacterial flagella. Again, differences in archaea. So we, instead of having flagella, we have an archaeolum in the archaea. So it's an analogous structure. It's oftentimes smaller, it's not hollow, and it's going to be powered by ATP, not the proton motive force. Again, not important necessarily to have all those details in your head, other than to know that they are different and that you could look them up if you needed to. But looking at flagella, long strand made of protein for motility, and they're going to spin. They spin like propellers, and their distribution, presence, or absence can be characteristic of a species. We have some special terms. Peritrichous, it means that you've got them spread around the surface of the cell. Polar monotrichous would be a different setup where you have a single flagellum at the end of the cell, and other arrangements as well are available. Again, I'm not too concerned about you picking up those specific words and having them, but though they can be fun to have, but to know that those differences are possible is what is your take home here. Now, if we look at this, it's kind of cool. Again, it's like tiny machines that have been built over billions of years of evolution. Super cool. Again, we have an anchor in the plasma membrane, whether you're uh, gram positive or gram negative. You're going to anchor this thing in the plasma membrane because you're going to move protons, a proton gradient across that membrane to power this thing. And literally, it's going to spin. What's really cool about this is when you start talking about flagellum, how do bacteria move that have them? And then you get to start to talk about chemotaxis, right? So the run and tumble is this classic phrase that we use for how bacteria that have flagellum move towards things that are beneficial to them or move away from things that are dangerous to them. So we talk about positive or negative chemotaxis, specifically chemicals, with plants or other things that have an interest in light. You talk about phototaxis or aphototaxis. And again, the more you use the language, the more you'll get a feel for it. But this idea of movement towards a chemical will be positive chemotaxis, up a perceived concentration gradient, or down it, depending on what you need. But this idea of run and tumble is cool. And I love this quote from this paper from 2010, The Hydrodynamics of a Run and Tumble Bacterium Propelled by Polymorphic Helical Flagella. It's a really intense paper with a lot of math, but this quote is great that explains this idea, I think, really well. When all rotary motors rotate in the same direction, the cell swims forward, i.e., that's our run state. The cell changes direction by reversing the rotation of at least one of the motors, a tumble state. So although a tumble reorients the cell in a random direction, it is able to migrate stochastically towards a more favorable environment by changing the frequency of the reorientation, depending on the gradient of temperature or concentration of molecules of interest, right? So that's a mouthful, but when you pair that quote with a picture like this, it makes a whole lot of sense. So now all of a sudden, if I've got bacteria that are not in a chemotaxis state, they're just like, there's nothing here that I'm interested in. I'm not interested in going any particular direction. I'm just going to run and tumble, run and tumble, run and tumble. And I'm going to just be moving around randomly. Why? Because I might run into something that is interesting. And when I do run into something interesting, I pick up one of those receptors on my plasma membrane that says, hey, there's something here that I want. I start to initiate that run and tumble movement, change some of those flagella. When I'm getting an increase in receptor binding, I keep a run state that's longer until I find an overall increase in the thing that I'm interested in. So it's just super cool because in my mind, it really emphasizes this idea of a programmable tiny machine. Like that's what life is. And the programmable components are these proteins, right? So that's something that you definitely should be aware of, this idea of run and tumble for how bacteria move towards things that they want, those bacteria specifically that have flagella. Now, as we continue with our survey of external structures, the pili then are something else that you oftentimes hear. Now, again, not every bacterium have pili, pilum for singular, but they're smaller, shorter than flagella. It's the same idea that I've got proteins that I've built into a long string thin rod, right? Now, fimbriae is another term that you'll hear associated with pili, and it's kind of like the common pili. So where pili would be a bigger term, fimbriae would be more specific. Oh, what am I talking about specifically? Oh, the common pili that you find around the cell. What are they for? Attachment. So think about bouncing a ball off the wall. Now put a bunch of spikes on the ball. You throw the ball, and now it sticks to the wall, right? That's the kind of idea with fimbriae. Fimbriae. At the same time, some of them may be associated with movement. So we'll talk about bacterial with a twitching motility or a smooth gliding. It's stuff you don't have to worry about, but these would be involved in that as well. Now, a very special type of pilus is called the sex pilus, and we'll talk about this more when we get into bacterial genetics. But this is literally going to be a hollow protein tube that is built like a pilus, that is going to connect the cytoplasm of two different cells. And what we'll talk about later is how then a bacterium will literally feed a copy of its DNA through that pilus into the other cell. So we talk about, kind of sexual reproduction being a combination or exchange of genetic material. That's where we call this as a sex pilus, because literally a bacterium is feeding another bacterium its genetic material that is going to change the gene and hence the genes of and hence the phenotype potentially of that recipient cell. So we talk about bacteria being F minus or F positive, whether they can form a sex pilus or not, right? So those are the things we want to think about as far as external components go. Internal components are going to be a much more kind of a laundry list, which I have here. We'll spend a little more time on endospores. But for inside the cell, we're going to think about the DNA. We know that prokaryotic cells have a single chromosome. It's circular. Eukaryotic cells have linear chromosomes and more than one. We're going to see some degree of packaging in the DNA for prokaryotes, but not nearly to the extent that we see in eukaryotes. But we still do have to package. We have DNA binding proteins, and that's where we get this idea of a nucleoid region inside the cytoplasm. We also are going to talk about plasmids to a great extent as we get into genetics. We'll deal with plasmids in the lab this quarter as well. So plasmids then are small independent pieces of circular DNA. You can have plasmids in eukaryotes as well. So we oftentimes will use plasmids with yeast if we want to mess around with yeast genetics and eukaryotic genetics. But it's very, very easy to do with prokaryotes and bacteria. So this is what I have a lot of experience with as a molecular biologist, building plasmids, modifying plasmids, transferring plasmids to bacteria, modifying, getting them to do the work for me by changing their genetics. And almost always we'll do that using plasmids, at least initially. You can use plasmids later then to actually change the genomic DNA of a particular prokaryote and kind of make it permanent, because plasmids can be lost or gained pretty easily. The other thing we want to think about internally with prokaryotes is their ribosomes. Again, differences are powerful. So the fact that prokaryotes have 70 S ribosomes as compared to the 80 S ribosomes of eukaryotes means that this is a potential target for antibiotics, and in fact it is. So ribosomes are ribosomes. Their job is the same. They're binding to an mRNA molecule and using that as a template to build proteins. But because the structure is slightly different, we can target those slight differences and provide a chemical that won't impact an 80S ribosome, but will knock out a 70S ribosome. And if you knock out protein production, you knock out a cell. The cytoskeleton for prokaryotes is mostly actin and tubulin. It's certainly not going to be as complicated as a eukaryotic cell, but you do have a cytoskeleton elements in there. They're going to be involved in cellular reproduction, binary fission, which we'll talk about a little bit later as well, and support for the cell, as all cytoskeletons do well. Storage granules are going to be present. There's a variety of forms based on what you're storing. So accumulation of high molecular weight polymers synthesized from a nutrient that a cell has a relative excess. You're like, hey, I got plenty, let's store it for later. These often become generic terms to a certain extent, especially when you're looking under a microscope. It's like, oh, look, I don't know what that is. It looks like a storage granule, right? And then also we have protein-based compartments. So the fact that prokaryotes are defined by not having membrane-bound internal compartments is saying one thing. It's not to say that they can't store stuff as well. Storage granules are naked, but protein-based compartments are going to have a protein coat that you can put stuff in, like a gas vesicle, to control the buoyancy of a prokaryote in the water column. Microcompartments and then encapsulating nanocompartments are literally like what you would see like on a viral level. As a viral head encapsulates its DNA, that is kind of the level of what we would think of as nanocompartments inside prokaryotes. Again, this is something that's still kind of your first microbiology course. These are things that you would go into much greater detail where you take more advanced and specific microbiology. The final thing I want to talk about in this class, or in this lecture rather, is endospores. So endospore generation is, again, diagnostic. So species that can do that, like some bacillus species or Clostridium species, it's important. And if they see endospores in a culture, oh, that can be point me towards those specific genus species. Now, the things that we're interested specifically that have endospores are the diseases that come from bacteria that cause botulism, tetanus, gangrene, pseudomembranous colitis, anthrax. That's the one that that I heard about first, and I think it's super interesting. So what is an endospore? It's going to be a cell that has come to a dormant state that is very resistant to the external environment. So resistant to heat, resistant to desiccation, resistant to chemicals, resistant to UV light, etc. So these are the things you have to like. So if you're dealing with a species that can form endospores, you have to be very particular about making sure that you are killing the endospores as well, unless you want to risk contamination. Sporulation is the name that we use for the process of forming an endospore, and it's not fast. So where we think about, under optimal conditions at least, bacteria are reproducing 20 or 30 minutes, endospores are going to be very deliberate. It's going to be an 8-hour development. We'll look at it, the basic components of it, but again, it's not something to worry about too much. When that endospore then breaks dormancy, we talk about calling that germination. So it's very plant kind of terminology, but that germination is the exiting from the dormant state, and you get vegetative cells developing. Those vegetative cells tend to be extra sensitive to their environment. It's like kind of a trade-off, but then they will start to go through binary fission and kind of come back to the steady state for the version of that prokaryotic cell. Now, looking at sporulation, it's kind of a fun process. Again, you don't necessarily need to know it in great detail, other than the fact that a cell is going to divide, and that one of those cells is specifically going to be designated as the future endospore, and that the other cell is going to literally engulf it and then start building the spore coat and the core wall around that now endospore. And again, what do we have there? We have something that can dry out, rehydrate later, will grow back. It's a great way to provide a particular species with kind of insurance. So if some percentage of a population is always in an endospore form, that if something comes through like the autoclave and tries to kill all the bacteria, the endospores can be like, okay, we're hanging out, and when things get better, we're gonna pop back, go vegetative, and we're on our way again. We've not been killed. So that's the idea of the endospore. And we'll talk about this more, and we'll do an endospore stain in the lab. It'll be great. So to kind of finish this one up, again, now we've got a great chart that we've kind of moved our way through, starting with the cytoplasmic membrane, talking about cell walls, external structures, internal structures. You should be able to kind of work your way through that and have something to say about all these things.